Hi everyone, this is Glenda Ganzon and welcome to my Human Anatomy and Physiology class. And for today's video, I'm going to discuss the anatomy of the nervous tissue. So stay tuned. Nervous tissue is composed of two types of cells, neurons and glial cells. Neurons are the primary type of cell that most anyone associates with the nervous system. They are responsible for the computation and communication that the nervous system provides. And they also are electrically active and release chemical signals to target cells. Glial cells or glia are known to play a supporting role for nervous tissue. Ongoing research pursues an expanded role that glial cells might play in signaling, but neurons are still considered the basis of this function. So neurons are important, but without glial support, they would not be able to perform their function. Neurons are the cells considered to be the basis of nervous tissue. They are responsible for the electrical signals that communicate information about sensations and that produce movements in response to those stimuli, along with inducing thought processes within the brain. An important part of the function of neurons is in their structure or shape. The three-dimensional shape of these cells makes the immense number of connections within the nervous system possible. As you learned from my previous discussion, the main part of the neuron is the cell or the cell body, which is also known as the soma, which means body. The cell body contains the nucleus and most of the major organelles. But what makes neurons special is that they have many extensions of their cell membranes, which are generally referred to as processes. Neurons are usually described as having one and only one axon, that is a fiber that emerges from the cell body and projects to target cells. That single axon can branch repeatedly to communicate with many target cells. It is the axon that propagates the nerve impulse, which is communicated to one or more cells. The other processes of neuron are dendrites, which receive information from other neurons at specialized areas of contact called synapses. And the dendrites are usually highly branched processes providing locations for other neurons to communicate with the cell body. So information flows through a neuron from the dendrites across the cell body and down the axon. This gives the neuron a polarity, meaning that information flows in this one direction. Where the axon emerges from the cell body, there is a special region referred to as the axon hillock. And this is a tapering of the cell body toward the axon fiber. And within the axon hillock, the cytoplasm changes to a solution of lipid components called axoplasm. Because the axon hillock represents the beginning of the axon, it also referred to as the initial segment. Many axons are wrapped by an insulating substance called myelin, which is actually made from glial cells. Myelin acts as insulation, much like the plastic or rubber that's used to insulate electrical wires. A key difference between myelin and the insulation on a wire is that there are gaps in the myelin covering of an axon. And each gap is called a node of Ranvier. And it is important to the way that electrical signals travel down the axon. The length of the axon between each gap, which is wrapped in myelin, is referred to as an axon segment. And at the end of the axon is an axon terminal, where there are usually several branches extending toward the target cell, and each of which ends is an enlargement called a synaptic end bulb. And these bulbs are what make the connection with the target cell at the synapse. There are many neurons in the nervous system, a number in the trillions, And there are many different types of neurons, and they can be classified by many different criteria. The first way to classify them is by the number of the processes attached to the cell body. Using the standard model of neurons, one of these processes is the axon and the rest are dendrites. Because information flows through the neuron from dendrites or cell bodies toward the axon, these names are based on neurons' polarity. The first is unipolar. Unipolar cells have only one process emerging from the cell. True unipolar cells are only found in the invertebrate animals, so the unipolar cells in human are more appropriately called as pseudo-unipolar cells. Invertebrate unipolar cells do not have dendrites. Human unipolar cells have an axon that emerges from the cell body, but it is split so that the axon can extend along a very long distance. At one end of the axon are dendrites, and at the other end, the axon forms synaptic connection with a target. So unipolar cells are exclusively sensory neurons and have two unique characteristics. First, their dendrites are receiving sensory information, sometimes directly from the stimulus itself. And secondly, the cell bodies of unipolar neurons are always found in ganglia. So sensory perception is a peripheral function, or those dendrites are in the periphery, perhaps in the skin. So cell body in the periphery, though closer to the central nervous system in the ganglion. The axon projects from the dendrite endings past the cell body in a ganglion and into the central nervous system. The next one is the bipolar. So bipolar cells have two processes which extends from each end of the cell body opposite to each other. One is the axon and one the dendrite. Bipolar cells are not very common and they are found mainly in the olfactory epithelium where smell stimuli are sensed and as part of the retina. The third one is the multipolar. So multipolar neurons are all of the neurons that are not unipolar or bipolar, and they have one axon and two or more dendrites, and they usually many more. With the exception of the unipolar sensory ganglion cells and two specific bipolar cells mentioned earlier, all other neurons are multipolar, and some cutting-edge research suggests that certain neurons in the central nervous system do not conform to the standard model of one and only one axon. So some sources describe a fourth type of neuron, and it is called an anoxonic neuron. The name suggests that it has no axon, but this is not accurate because anoxonic neurons are very small, and if you look at through a microscope at the standard resolution used in histology, you will not be able to distinguish any process specifically as an axon or a dendrite. So any of those processes can function as an axon depending on the condition at any given time. Nevertheless, even if they cannot be easily seen and one specific process is definitively the axon, these neurons have multiple processes and are therefore multipolar. Neurons can also be classified on the basis of where they are found, who found them, what they do, or even what chemicals they use to communicate with each other. And some neurons are named on the basis of those sorts of classification. For example, a multipolar neuron that has a very important role to play in a part of the brain is called the cerebellum, is known as the Purkinje, commonly pronounced as Purkinje cell or Purkinje cell. It is named after the anatomist who discovered it. who is John Evangelista Perkinji. It was discovered in 1787 to 1869. Glial cells or neuroglia or simply glia are the other type of cell found in nervous tissue, and they are considered to be the supporting cells, and many functions are directed at helping neurons complete their function for communication. The name glia comes from the Greek word that means glue and was coined by a German pathologist, Rudolf Virchow, who wrote in 1856. This connective substance, which is in the brain, the spinal cord, and the special sense nerves, is a kind of glue, or what we call as neuroglia, in which the nervous elements are planted. So today, research into nervous tissue has shown that there are many deeper roles that these cells play, and research may find much more about them in the future. So there are 6 types of glial cells, and 4 of them are found in the central nervous system and 2 are found in the peripheral nervous system. One cell providing support to neurons of the central nervous system is the astrocyte, so named because it appears to be star-shaped under a microscope. Astrocytes have many processes extending from their main cell body, not axons or dendrites like neurons, just cell extensions. And those processes extend to interact with neurons, blood vessels, or the connective tissue covering the CNS that is called the pia mater. Generally, they are supporting cells for the neurons in the central nervous system, and some ways in which they support neurons in the central nervous system are by maintaining the concentration of the chemicals in the extracellular space, removing excess signaling molecules, and reacting to tissue damage, and contributing to the blood-brain barrier, or this is what we call as the BBB. The blood-brain barrier is a physiological barrier that keeps many substances that circulate in the rest of the body from getting into the central nervous system, restricting what can cross from circulating blood into the central nervous system. So nutrient molecules such as glucose or amino acids can pass through the BBB, but other molecules cannot, and this actually causes problems with the drug delivery to the central nervous system. Pharmaceutical companies are challenged to design drugs that can cross the BBB as well as have an effect on the nervous system. So, like a few other parts of the body, the brain has a privileged blood supply, and very little can pass through by diffusion. Most substances that cross the wall of a blood vessel into the CNS must do so through an active transport process. Because of this, only specific types of molecules can enter the CNS. Glucose, which is the primary energy source, is allowed, as the amino acids. Water and some other small particles like gases and ions can enter also, but most everything else cannot, including white blood cells, which are one of the body's main lines of defense. While this barrier protects the CNS from exposure to toxic or pathogenic substances, it also keeps out the cells that could protect the brain and spinal cord from diseases and damage. So the BBB also makes it harder for pharmaceutical to be developed that can affect the nervous system. Aside from finding efficacious substances, the means of delivery is also crucial. Also found in the CNS tissue is the oligodendrocyte, sometimes called just oligo, which is the glial cell type that insulates axon in the central nervous system. The name means cell of few branches, and there are few processes that extend from the cell body and each one reaches out and surrounds an axon to insulate it in myelin. And one oligodendrocyte will provide the myelin for multiple axon segments, either for the same axon or for separate axons. Microglia are smaller than most other glial cells. Ongoing research into these cells, although not entirely conclusive, suggests that they may originate as white blood cells called macrophages that become part of the CNS during early development. So while this origin is not conclusively determined, whether function is related to what macrophages do in the rest of the body. So when macrophages encounter diseased or damaged cells in the rest of the body, they ingest and digest those cells or the pathogens that cause disease. Microglia are the cells in the central nervous system that can do this in normal healthy tissue, and they are therefore also referred to as CNS resident macrophages. The ependymal cell is a glial cell that filters blood to make cerebrospinal fluid, or CSF, the fluid that circulates through the CNS. Because of the privileged blood supply inherent in the BBB, the extracellular space in the nervous tissue does not easily exchange components with the blood. Ependymal cells line each ventricle and one of four central cavities that are remnants of the hollow center of the neural tube formed during the embryonic development of the brain. The choroid plexus is a specialized structure in the ventricle where ependymal cells come in contact with blood vessels and filter and absorb components of the blood to produce cerebrospinal fluid. Because of this, ependymal cells can be considered a component of the BBB, or a place where the BBB breaks down. And these glial cells appear similar to epithelial cells, making a single layer of cells with little intracellular space and tight connection between adjacent cells. They also have cilia on their apical surface to help move the CSF through the ventricular space. One of the two types of glial cells found in the PNS is the satellite cell. Satellite cells are found in sensory and autonomic ganglia where they surround the cell bodies of the neurons. This accounts for the name based on their appearance under the microscope, and they provide support performing similar functions in the periphery as astrocytes do in the central nervous system, except, of course, for establishing the BBB. The second type of glial cell is the Schwann cell, which insulate axons with myelin in the periphery. So Schwann cells are different than oligodendrocytes in that a Schwann cell wraps around a portion of only one axon segments and no others. Oligodendrocytes have processes that reach out to multiple axon segments, whereas the entire Schwann cell surrounds just one axon segment. The nucleus and cytoplasm of the Schwann cell are on the edge of the Myelin Sheath. The insulation for axons in the nervous system is provided by glial cells, oligodendrocytes in the CNS, and Schwann cells in the PNS, whereas the manner in which either cell is associated with the axon segment or segments that it insulates is different. The means of myelinating an axon segment is most the same in the two situations. Myelin is a lipid-rich sheath that surrounds the axon, and by doing so creates a myelin sheath that facilitates the transmission of electrical signal along the axon. The lipids are essentially the phospholipids of the glial cell membrane. Myelin, however, is more than just the membrane of the glial cell, and it also includes important proteins that are integral to that membrane, and some of the proteins help to hold the layers of the glial cell membrane closely together. So the appearance of the myelin sheath can be thought of as similar to the pastry wrapped around a hot dog or pigs in a blanket, or a similar food. The glial cell is wrapped around the axon several times with little to no cytoplasm between the glial cell's layers, and for oligodendrocytes, the rest of the cell is separate from the myelin sheath as a cell process extends back toward a cell body. A few other processes provide the same insulation for the axon segments in the area. For Schwann cells, the outermost layer of the cell membrane contains cytoplasm, and the nucleus of the cell as a bulge on one side of the myelin sheath. And during development, the glial cell is closely or loosely or incompletely wrapped around the axon. The edges of this loose enclosure extend toward each other, and one end tucks under the other. The inner edge wraps around the axon, creating several layers, and the outer edge closes around the outside so that the axon is completely enclosed. Myelin sheaths can extend for 1 or 2 millimeters depending on the diameter of the axon. Axon diameter can be as small as 1 to 20 micrometers because a micrometer is 1/1000 of millimeter, and this means that the length of the myelin sheath can be 100 to 1000 times of the diameter of the axon. So this ends my discussion about the nervous tissue. If you have any question, please write it in the comment section down below and I'll be glad to answer them all. And if you're still not subscribed to my channel, please also do consider subscribing and hit the notification bell so that you'll be updated with my new videos. And if you like this video, please give it a thumbs up and share. So once again, I would like to thank you all for watching my video and for listening to my discussion. Until my next video, bye everyone!